and today we will discuss two of those cases that do not represent the reaction velocity profile in terms of hyperbolic kinetics. One of them is substrate inhibition, the other is sigmoidal kinetics, particularly as it applies to multi-site and allosteric enzymes. Now first we come to now in fact if you notice that these two kind of things, if you look into the reaction profile, a typical substrate versus initial reaction velocity profile for a hyperbolic kinetics goes like this with a asymptotic reaction velocity Vm. Now in the case of substrate inhibition, essentially the reaction velocity when you increase substrate concentration does not reach to the saturation level, rather even earlier than that it deviates and passes through a maximum. The reaction velocity when substrate concentration goes high, the reaction velocity starts dropping and does not retain at the saturation level. On the other hand, in the sigmoidal kinetics, instead of a reaction profile being represented by a rectangular hyperbola, it is in the form of a sigmoid. That is, of course ultimately it can reach to the same kind of saturation behavior, but the initial profile will be different in the case of a sigmoidal profile, and these two different situations will give you an idea that in some cases, and these are very important cases and have lot of physiological importance for the cell, to have designed such enzymes which deviate from hyperbolic kinetics, as we will see further. Now coming to the first case of substrate inhibition, as I mentioned earlier, the reaction profile passes through a maxima, and at a particular substrate concentration it essentially reaches maximum value and then start dropping. Now this substrate concentration essentially is a substrate concentration corresponding to maximum reaction velocity. Of course this substrate concentration corresponding to maximum reaction velocity is not really something which is corresponding to maximum reaction velocity of enzyme catalyzed reaction. It is as a result of certain enzyme substrate interactions such that the rate becomes tapering, rate gets tapered even before the saturation level reaches. Now of course this effect can also be visualized when you look for the classical Lineweaver plot. Instead of giving you a straight line, it becomes convex to the x axis and it can meet, and that can give you an indication that the enzyme catalyzed reaction is having some kind of a substrate. There are a number of examples in practice for such kind of a behavior in the reaction. I think one of the most conventional and simplest example could be the hydrolysis of sucrose to glucose and fructose by invertase, a very classical example where at a substrate concentration greater than 0.1 molar, I mean 0.1 molar is not a very high substrate concentration, is about say 3.4 or 3.5%. Even at that level the effect of substrate inhibition is noticed and the reaction velocity starts dropping. Another example of substrate inhibition is in the form of succinate dehydrogenase, which catalyzes the reaction interconversion of succinate to fumarate, essentially a reaction which catalyzes the formation of a double bond. Now succinate as a substrate has two carboxylic groups. As per the normal function of the enzyme, the two carboxylic groups of the substrate are required to be bound to the enzyme molecule. Now when substrate concentration becomes very high, the possibility or the statistical feasibility of the one carboxylic group from each of the two substrate molecule binding to the same enzyme molecule becomes much higher, and but in that process when the two substrate molecule get bound to one enzyme molecule and one carboxylic group of each of the substrate molecule, then the reaction is not catalyzed and ultimate enzyme substrate complex is a dead end complex.
So explain this.
As I mentioned earlier, Now, succinic acid or if you just look at the structure of succinic acid, it is essentially COO, CS2, CS2, CH, COO. Now, this is succinate. Now, essentially as per the normal reaction of succinic dehydrogenase, the substrate molecule, the 2 carboxylic group of substrate molecule are required to be bound to the enzyme active sites, and at low concentration of substrate this can very well happen. Now, when the substrate concentration becomes very high, it might end up that the enzyme molecule might bind to 2 substrate molecules, one of the carboxylic group from each of the succinic acids. So, therefore, the active sites are not functional yet, because instead of one molecule there are 2 molecules attached, and the enzyme substrate complex so results in the form of a dead end complex which does not go into the final product forming stage. Now, for such a reaction there is a feasibility that now essentially you may end up in a enzyme molecule having such kind of a conformation instead of having the 2 carboxylic group of the same enzyme, same substrate molecule bound to the enzyme, there are 2 substrate molecules bound to the same enzyme and 1 carboxylic group of each of the substrate molecule, and such a complex essentially does not form the product. Now, in such a situation can be explained, and of course, similarly in the case of hydrolysis of sucrose by invertage also, by one of the plausible mechanistic model that when one substrate molecule binds, the enzyme substrate complex leads to a product formation. If the enzyme is bound to 2 substrate molecule, a dead end complex is formed. A simple reaction mechanism can be expressed like ES going to E plus P, then plus S going to ESS. Now, essentially we can write exactly in our nomenclature of K1, K minus 1, K2, and this equilibrium of the second substrate binding can be expressed, let us say, by dissociation constant of Ks, individual rate constant being K small ks and K minus S. Now, this ESS essentially is a dead end complex. That means the product formation will take place from this substrate complex. This will not lead to any product formation, and whatever fraction of the total enzyme is bound in the form of ESS will not participate in the reaction, with the result that your reaction velocity will drop. That means at the saturation level you are assuming that all the enzyme is bound to the substrate. So, essentially all the enzyme is not bound to the substrate. Here it starts falling, more of the enzyme at increasing the substrate concentration leads more of ESS formation, and therefore now based on this, on the equilibrium, on the basis of your equilibrium kinetics, one can write the rate expression of this such a system as Bi over Bm is equal to S upon Km plus S 1 plus S upon Ks. Now, as I mentioned earlier, the Vm is the true saturation velocity, to maximum reaction velocity if there was no such substrate inhibition to take place, and Km is the classical K minus 1 plus K2 upon K1, the Michaelis-Menten constant, and Ks is the dissociation constant of the second substrate binding. Now, in such a case, if you notice, and then your this also can be written as your VIS is equal to Vm into S upon Km plus S square upon Ks. Now, if you plot such profile, the rate expression will pass through a maxima, and if you differentiate that is your dV over ds and equate it to 0, you will end up with maxima, and the maximum reaction velocity will be obtained at a substrate concentration which is square root of Km into Ks. One can develop the expression to write the differential and equate it to 0, and that will end up in the maximum, the substrate concentration for the maximum reaction velocity, that is the maxima at the. So, in such cases you notice that the such enzymes do not really strictly follow the typical Michaelis-Menten kinetics. There are now another group of enzymes, as we listed in our earlier sheet, which are essentially multi-site and rather multi-site. Now, large number of enzymes, as we noticed right in the beginning, that the chemical nature of the enzyme provides for a quaternary structure in some cases, and the quaternary structure is composed of multiple subunits arranged in a particular fashion, so that their interaction brings in a conformation, thereby active sites are created. Now, these multi-site enzymes essentially are oligomeric in nature, which consist of more than one subunits, and each subunits very often, now these subunits might be identical. They also may have, each subunit may have a active site for enzyme catalyzed reaction, multiple active sites per enzyme molecule. And if now there are two kind of situation, one is that these active sites on these oligomeric enzymes might be identical and independent. Now, notice the two terms which I have highlighted here. Identical means the catalytic sites have identical free energy change of binding. That means the free energy of binding on either side is same, and essentially it does not matter to which site the substrate molecule binds first or second, it makes no difference. They are identical in terms of conformation. The independent subunits or the independent binding sites implies essentially that binding to one site does not bring in any conformational change so as to change the affinity of other binding sites. Very often it might happen that initially the binding sites may be identical and they are free for the substrate to bind to any one of them. Once the substrate binds to one of the binding sites, in some cases the binding site might induce a conformational change, thereby making the affinity of the substrate molecule to the other binding sites more favorable or sometimes unfavorable, of course. But when we talk of allosteric enzyme, we are talking of a favorable interaction. So, in the simple case of multi-site enzymes is identical independent subunits. Now, in such cases when all the binding sites or all the catalytic sites on the enzyme molecule, even located at different subunits, if they are identical and non-interacting or non-cooperative, I mean they have no interaction among each other, the net result will be that if you are handling something like, if you consider that let us say 1 nanomole of an enzyme substrate concentration of n binding sites per mole will be equal to n picomole of one binding site molecule. Basically the rate profile will be hyperbolic in nature, as we mentioned earlier, and the only difference is that the maximum reaction velocity term in that case will be n into k2 into Et. That means our maximum reaction velocity in the case of a single binding site is Vm is equal to k2 into E naught. If there are n binding sites, the only difference will be it will become n into k2 into E naught, because all the n binding sites have to be saturated. Except with that difference, there will be no difference and the reaction profile will be hyperbolic in nature, and one can examine such a situation in the case of a variety of identical independent subunits, which essentially, for example, just to illustrate my point with a simple example of a dimer, in the case I mean having two identical binding site, a simplistic case. Now the reaction velocity, the reaction mechanism can be written as Es going to E plus P, that is your, let us say this is the diffusion constant Ks. Now the enzyme can bind to another site also, forming Se, here also it is Ks, then of course plus S, the second binding site SES and plus S. Now, in all the 4 cases of equilibrium, the diffusion constant is Ks, identical. That means all the binding sites are equal, they do not influence the binding to the other side, whether the first binding site is occupied, the second binding site is free, and depending on the substrate concentration it can bind and form a binary complex where the 2 substrate molecules are bound. That means both the binding sites. The only difference will be when SES gives you a product, the rate of product formation will be 2 K2. This is K2, this is 2 K2, because and it will lead to your ES plus P and SE plus P essentially. And similarly here also you can have a product formation to P plus E. So, you can have from either direction the product formation and they are independent. I mean similar situation might arise for a tetramer or a hexamer depending on the number of binding site on the enzyme molecule. Now, just for such a simple case, the rate expression which can be written as S upon Ks plus S square upon Ks square whole divided by 1 plus 2S upon Ks plus S square upon Ks square. Now, here we must yes.
SE and ES.
You see, if you consider that there is an enzyme molecule, it has binding sites on both the directions, on both the sides. Now, S and ES is difference only. S represent one active site and ES represent the other binding site occupied. That means essentially that whether it binds on the left hand side or on right hand side, the binding of the next substrate onto the vacant site has no effect.
And so the SES converts to ES plus P or SE plus
either SE plus P. It can again from there both the binding sites can dissociate independently and lead to the formation of a single enzyme substrate complex plus product. And again, so therefore, I mean that means that means all the seats, let us say in the class, are totally vacant. Anybody who gets into the room can occupy any of the seat, and as many number of students can come in, as long as the number of seats are available they can occupy. And in such case the rate expression, only difference here we must notice is that this Vm term will correspond to essentially 2 K2 into E naught rather than yes.
So can't SES lead to formation of 2 products in one enzyme, E plus 2P?
Eventually it will lead to that, yes. Eventually, that is why we are saying Vm is 2K. If suppose whole of the enzyme is in this form, whole of the enzyme is in SES form, both the sides are totally saturated, and eventually ultimately because ES and SE also will lead to the product formation. So, the net effect will be that SES is giving you 2 product and the free enzyme essentially, that is true. And the only difference is here the definition of Vm must be taken as 2 K2 into, or in other words, if it is let us say a multimeric protein or enzyme where n binding sites are there, this can become n into K2 into E naught. Otherwise, the treatment can be identical as in the case of a hyperbolic profile. Now, this also, as you notice, or in general you can write your V upon Vm will be S upon Ks 1 plus S upon Ks n minus 1 upon 1 plus S upon Ks to the power n. Now, this is if you just look at extrapolate your number of binding sites from 2 to 4 to 6 to n. Now, this is the kind of rate expression you end up, and this can be this is equal to upon S upon Ks upon 1 plus S upon Ks, that is equal to S upon Ks plus S, which is essentially, as you notice, is same as hyperbolic rate expression. So, in fact, when there are n number of binding sites, the only difference, as I mentioned, is that the definition of Bm will change, rest of the profile of the rate profile will remain identical, and there will be no difference. Now, in the case of in the second and more complex case, when we talk of cooperative binding, that means the binding sites present on the enzyme molecule, let us say if you consider two binding sites on the enzyme molecule, they are not, they may be identical. That means the free energy change for the binding process may be same, and that is what we mean essentially by the identical binding sites. That means to start with, the substrate molecule can hook up to any of them irrespective of the choice, but after it gets bound, it makes a conformational change on the enzyme molecule. That means it influences the conformation of the other binding sites, therefore altering its affinity.
So do you mean to say it's a step by step binding?
Yes, in fact there are number of sort of mechanistic hypotheses which have been proposed, we will come to that, but it is essentially yes, the first binding site is free for all the substrate molecule, and once this is bound, if it makes a change on the conformation of the other binding sites such that the affinity of the substrate to the binding site gets altered, obviously if we want a positive a favorable effect, the affinity must increase or the Km value must decrease, dissociation constant must decrease. Now, in that case, if the Km value of the subsequent binding sites decreases with the binding of the first substrate molecule, we call it cooperative binding. Now, essentially the significance of cooperative binding lies in actual practice in the case of allosteric enzymes, and you all know probably with your background in the biochemical metabolism that in each metabolic pathway you have certain regulatory enzymes which behave in a allosteric manner. By allosteric manner I mean that the binding substrate binding process in the case of those enzymes is cooperative in nature, and essentially then they do not exhibit the typical hyperbolic kinetics, rather they exhibit a sigmoidal kinetics. And if you notice a sigmoidal kinetics, if you just compare the two kind of profiles, in the case of a sigmoidal profile, this is a sigmoidal profile, this is a hyperbolic profile. It is for a hypothetical two different enzymes which are coincidentally having the same velocity at substrate concentration, let us say some 9 arbitrary units. Now, in the case of both of the cases, whether it follows a hyperbolic kinetics or a sigmoidal kinetics, the enzyme velocity reaches to the same value at a particular substrate concentration. That is what we are talking there at the maximum reaction velocity. Let us say if subsequently it reaches enough, but only initial phase changes, and the first one is a hyperbolic typical profile, the second one is a sigmoidal profile which is exhibited by allosteric enzyme. Now, you will notice that the allosteric behavior has in some way an advantageous situation for the regulatory enzymes to function. Essentially, the role of regulatory enzymes in a metabolic pathway is the control of the metabolic pathway. That means it should be able to switch on and off the pathway with a very subtle change in substrate concentration. Now, for example, as we will see now, if you notice that if we notice here, let us say in these two profiles which have identical reaction velocity at a high substrate concentration, but if you notice that for the two enzymes to act, if they go from 0 to 3 substrate concentration, let us assume that in the early phases when the substrate concentration increases from 0 to 3 arbitrary units, whatever are the units, you will notice that in the case of sigmoidal kinetics the reaction velocity is only 0.1 again some arbitrary units, whereas in the case of a saturation kinetics or hyperbolic kinetics the reaction velocity can reach to almost 7.75 75% of the saturation value, a very high level. That means at a very small change of concentration the reaction velocity can be very high. But on the other side, if you take let us say two different the substrate concentrations required for two different levels of activity to reach, for example say between 0.75 and 0.1 substrate concentration for reaction velocity to reach 0.75 and 0.1. In the case of saturation kinetics or hyperbolic kinetics, the concentration of this ratio, the ratio of the concentration of substrates for the two levels to reach will be almost about 27 times. That means at 0.1 it can reach only at a very small substrate concentration. Here the ratio is 27 fold. On the other hand, in the case of sigmoidal kinetics this ratio is only 2.3. That means at a very small concentration change, normally what happens in the case of living cells, the intercellular concentration of substrate does not change over a very large range. I mean there will be hardly any situation where a substrate concentration can change 27 fold or such a high level of change, whereas it will for control purpose it will require a very small sort of a concentration change by which the reaction velocity can be regulated, and that is what can happen in the case of a sigmoidal kinetics. That means just by changing almost a concentration ratio of 2.3 fold, 2 to 3 fold, you can notice that the reaction velocity can be altered by from 0.1 to 0.75, and that is the advantage with this kind of behavior, the reaction behavior, the regulatory enzymes obtain in the case of cellular metabolism. And with this kind of a kinetic behavior, the enzymes are able to act as almost like an on and off switch. That means the substrate concentration comes down, reaction velocity is very slow, the metabolic pathway is slowed down, it is not made to 0, or if the substrate concentration slightly increases, the pathway is fully on and the product formation or whatever the corresponding intermediates in the pathway are fully available, and that advantage it provides to the cellular metabolism. Now such a behavior of sigmoidal kinetics has been very appropriately explained at least by two mechanistic hypotheses. One, and probably in its simplest form, is on the basis of sequential interaction model. Now this is essentially an extension of Coastlands induced with hypothesis, which we discussed when we were talking about the specificity, how the enzymes obtain their specific action. We remember that we talked about the Coastlands induced with hypothesis, whereby when the substrate molecule binds to one of the binding sites, the enzyme undergo a conformational change so as to fit onto the substrate molecule. This is something like an extension to that, and which essentially means that when the substrate molecule binds on one of the binding sites of the enzyme, it immediately triggers off a conformational change, whereby the affinity for the next binding sites becomes much higher, and they expressed it with the interaction factor, that for each binding of the substrate to a binding site, the affinity reduces by a factor, let us say alpha, as an interaction factor. And that is what, if you notice in a diagrammatic picture like here, if this is a free enzyme which has two binding sites on identical binding sites, the substrate molecule binds, let us say, first to A, it immediately brings in a conformational change on the second binding site, whereby the dissociation constant reduces to alpha Ks. In fact, the conformational change leads to the dissociation constant or Michaelis-Menten constant for the second binding site, and then the second binding site also gets bound. Now, obviously, you will notice that it is logical that if this dissociation constant is low, will mean that the substrate will have much more affinity for the second binding site. And similarly, I mean, if it initially it binds with the Ks onto other binding site, it can influence the conformational change on the other binding site, and it can meet both the binding sites are occupied. And I mean, such a system can be represented by a mechanistic model, whereby you have the first binding as Ks either here or here. The second binding process is represented by dissociation constant of alpha Ks either here or here. The product formation with Es will be E plus P. Similarly, when both the binding sites are occupied, the product formation rate will be twice the Kp, because then it can go to either Se or Es, there is no difference. Now, in such a case, you will ultimately end up with the kinetic equation or kinetic expression, which will be expressing your the sigmoidal reaction velocity profile response. Now, obviously, the situation which I have shown here is rather too simplistic. That means, we have tried to explain with the two binding sites. In many cases, particularly with the regulatory enzymes, the number of binding sites may be 4 or 6 or 8, even tetramers are a very common example in the literature, and therefore, the rate expression will be quite complex, and their analysis can become very difficult. Now, to simplify the situation, what we there is an equation available corresponding to your Michaelis-Menten equation, which can be applied to allosteric enzymes. In the case of allosteric enzymes, we have a simplified rate equation which can express or which can describe the behavior of the rate behavior of the allosteric enzymes, and basically this was given by Hill. The basic assumption here is, now one of the basic assumption here which he has made is that these interaction factors, what I mentioned earlier, your Ks and alpha Ks, the ratio alpha is very small than 1. That means, the cooperativity is very high. Once the substrate molecule binds to one of the binding sites, it makes a drastic conformational change, so that the affinity is increased very significantly. Now, if you consider, let us say, the n binding sites, what will happen in such a situation, that with each binding site being occupied by the substrate, the next subsequent binding sites will have much more affinity. Ultimately, at equilibrium or under steady state condition, the net effect will be that the bulk of the enzyme will be in the form of ESN. Now, obviously, theoretically at equilibrium, the enzyme must be available in the form of E, ES, ES2, so on ESN. Now, normally, if there are n binding sites and the binding process continues sequentially, then the enzyme must be available in different species in different forms as E, ES, ES2, ESN up to ESN. But if the interaction factor alpha is very small, that means at each stage affinity is increased by a very significant level, or what we understand as cooperativity is very high. In that case, the assumption by Hill was that most of your enzyme will exist in the form of ESN. Most of the enzyme will exist. Of course, there will be small concentrations, but they can be neglected. And that based on that assumption, then he gave a rate expression which is B upon V max equal to S to the power n upon K dash plus S to the power n. Now, this is called your Hill's equation. Now, where n is the number of binding sites per enzyme molecule, and K naught is the Hill's constant. Now, mind it that this K naught is different than the Michaelis-Menten constant. very significantly different. Now, essentially as you know that Michaelis-Menten constant essentially is the substrate concentration at which your reaction velocity is 50% of that of the maximum reaction velocity. Now, that situation will not apply here if you analyze, and now if you put n is equal to 1, then the equation will get into Michaelis-Menten kinetics.
What is Vm here, sir?
Pardon?
Vm.
Now, Vm here is the maximum reaction velocity again when all these binding sites are occupied by the substrate. When all the binding sites are occupied by the substrate, that means I mean the substrate concentration is high enough, but again this is not identical to the Vm of the Michaelis-Menten equation, because in the case of Vm in the Michaelis-Menten equation, our Vm is a asymptotic value. You can never approach it practically. It is a theoretical value, whereas in this case of Vm you can approach to that value when all the substrate is, all the enzyme binding sites are totally occupied by the substrate molecule. Now, if you look at, if you compare this K naught with the Michaelis-Menten constant, now when V is equal to 0.5 Vm, I mean if you just take the same sort of yardstick of explaining K naught as we take in the case of Michaelis-Menten equation, V is equal to 0.5 Vm, then essentially you will end up as S 0.5 to the power n is equal to K naught in the case of Hill's equation, or your S 0.5 substrate concentration for 50% of the maximum reaction velocity is nth root of K naught, and therefore, and there it was half of the maximum reaction velocity. So, I think that brings in a distinction between the two parameters that we are talking about. Now, essentially the level of sigmoid, that means the level of cooperativity for different enzymes, let us say how much it deviates from the hyperbolic performance, or how much it is more efficient in terms of regulatory behavior. You know, as we mentioned when we talked about the ratio of the two substrate concentration for two levels of reaction velocity, the smaller it is more regulatory, it is a better regulatory system. It is a very sharp change in substrate concentration, it can bring in the regulation. And so, in that case, now similarly one can express the cooperativity index of any given allosteric enzyme by expressing it in terms of the ratio of substrate concentration for achieving two different levels of reaction velocity. For example, if you consider, I mean just as a practice, as a conventional practice, we take the two reaction velocities at 0.9 Vm and 0.1 Vm, that means 10% and 90% of the, and the ratio of the two S 0.9 upon S 0.1 is conventionally called as cooperativity index. Of course, this is just I mean kind of a conventional practice. You can define your cooperativity index as long as you mention if it is S 0.75 upon S 0.1 or S 0.9 upon S 0.1, it could be any ratio can be used to express cooperativity index. It is only a matter of convention that we are following the ratio of at 90% and 10% values. Now, essentially if you put down the values, if you just get the value of S 0.9 and S 0.1 in your original Hill's equation, that is your V upon Vm is equal to S to the power n upon K dash plus S to the power n, your S 0.9 will be equal to nth root of 9 K dash, and S 0.1 will be equal to nth root upon Ks upon 9, and the ratio essentially cooperative index will be nth root of 81.
K prime
sorry, you are right, k prime upon 9, and the cooperative index, therefore, the ratio will be nth root of 81, or in other words the n will be equal to log 81 upon log s 0.9 upon s 0.1. So, therefore, n is the number of binding sites, and therefore, one can determine, if one can determine the value of s 0.9 and s 0.1, the ratio, one can determine the number of binding sites. Now, this number of binding sites n, again experimentally, theoretically it should be a integer, the number of binding sites will be 1, 4, 2, 3, 5, any integer, but practically if you notice, you will never get a integer value, you will get a fraction, say 3.4 or 3.6, and essentially the reason for that is the basic assumption in the Hill's equation, that is the all the enzyme at equilibrium will be in the form of ESN, whereas there will be small concentration of all other enzyme species, that means with the partly bound enzyme the substrate bound forms, and so therefore, the final value of n will be in the fraction, and as a matter of conventional practice, if we get let us say 3.6, we consider the next integer, that probably the binding sites are 4, because the number of binding sites which we guess less is primarily because of the other not completely saturated enzyme molecule. If you also look at the Hill's plot, just to, if you want to plot this expression, you get s to the power n upon k dash plus s n. You can simplify this to get your n log s plus log V m minus V upon V is equal to log k naught. Or log V upon V m minus V equal to n log s minus log k naught. Now, essentially if you make a plot, a log plot of log s versus log V upon V m minus V, you must end up in a straight line with the slope of n. And obviously, on a log plot, you can straight get a slope and get the number of binding sites. Now, also if you notice that at a value of log s corresponding to the value of log V upon as 1, if you put V upon V m minus V as 1, the log will be 0. In the log form it will be 0, of course. I am putting V upon V m minus V as 1, and at that value the corresponding value of log s will be log s 0.5. That means, if you recall, the log s 0.5 what we deduced earlier from our equation was n log s 0.5 is equal to log k naught. So, if you can know slope and determine the value of n and also determine s 0.5 log s 0.5, that means the substrate concentration for 50% of the maximum reaction velocity, you can get the value of k naught, what is known commonly as Hill's constant. And so, that gives you a neat analytical procedure to define your kinetic behavior of the allosteric enzymes that are available in the living systems, mostly for regulatory purposes.